Welcome to part 3 of our video on cell, the building block of life. We will continue our exploration of cell structure and function. The cell interior is a coordinated working system with 3 basic parts: the plasma membrane, cytoplasm, and a prominent nucleus. The cytoplasm contains subcellular components called organelles, visible with an electron microscope. This diagram illustrates the structure of a typical bacterial cell, a typical plant cell, and a typical animal cell. Here we see more detailed diagrams of plant and animal cells, highlighting various organelles and structures within them. This table compares the presence of key cell structures across bacterial, plant, and animal cells. Cells without a well-defined nucleus and membrane-bound organelles are prokaryotic cells. Plant and animal cells with a well-defined nucleus and membrane-bound organelles are eukaryotic cells. This table compares the characteristics of prokaryotic and eukaryotic cells, including the nucleus type, cell diameter, and cell number. Beyond the cell, we find viruses, viroids, and prions. They are infectious agents differentiated by their biological composition. Viruses, viroids, and prions are acellular infectious agents. Viruses have genetic material and a protein coat. Viroids lack a protein coat, and prions are misfolded proteins lacking genetic material. In eukaryotic cells, the cytoskeleton, a network of fibers, provides structural support, maintains cell shape, and enables movement. The cytoplasm may also store substances like starch or calcium oxalate, known as cell inclusions. Cell organelles in eukaryotic cells carry out various life processes simultaneously and independently. They build new materials, remove waste, and provide energy, acting together like a tiny living factory. The nucleus is a vital organelle containing the cell's genetic material. These diagrams show its key components. The nucleus includes the double-layered nuclear membrane with nuclear pores, the nucleolus, nucleoplasm, and chromatin material. Nuclear pores facilitate material transfer between the nucleus and cytoplasm. The nucleolus is where ribosomal subunits are synthesized, which then exit to the cytoplasm to form ribosomes. Chromatin material forms chromosomes when a cell is about to divide. Chromosomes are composed of DNA and specific proteins. Functional segments of DNA containing genetic information are called genes. Red blood cells, or RBCs, in humans are enucleate, meaning they lack a nucleus. This absence provides more space for hemoglobin, allowing them to transport a larger amount of oxygen. Their lifespan is about 120 days. Prokaryotic cells do not have a well-defined nucleus. Their DNA is a single circular molecule in a region called the nucleoid, as illustrated in this diagram of a prokaryotic cell. Ribosomes are tiny structures, often called the protein factories. They can be found freely in the cytoplasm or attached to the endoplasmic reticulum. Their primary function is protein synthesis. The endoplasmic reticulum, or ER, is a large, network-like organelle continuous with the nuclear envelope. It is a manufacturing factory, key in synthesizing and transporting proteins, fats, and some hormones. This image shows the structure of the endoplasmic reticulum, highlighting the distinction between the rough ER and the smooth ER, and their proximity to the nucleus. There are 2 types of ER. Rough ER, or RER, is studded with ribosomes and is involved in protein synthesis and secretion. Smooth ER, or SER, lacks ribosomes and is involved in the synthesis and storage of fats and hormones, as well as detoxifying cells. The Golgi apparatus is another crucial organelle, often referred to as the packaging and shipping center of the cell. The Golgi apparatus consists of stacks of flattened sacs and is functionally linked to the ER and other organelles. Its role is to modify, sort, and package proteins and lipids into vesicles for transport, secretion, or lysosome formation. The Golgi apparatus was first observed in 1898 by Italian scientist Camillo Golgi in the nerve cells of a barn owl, and later named in his honor when seen clearly under an electron microscope. Lysosomes are the cell's cleanup system. They are single, membrane-bound sacs filled with enzymes. These enzymes break down unwanted molecules and damaged cell parts, keeping the cell healthy. The resulting products are often reused in other cellular processes. Human sperm cells contain lysosomal enzymes that help break down the outer layer of the egg, enabling fertilization. Mitochondria are famously known as the powerhouse of the cell. Mitochondria supply the energy needed for most cellular activities. They have a smooth outer membrane and a folded inner membrane with finger-like projections called cristae, increasing the surface area for energy production. During cellular respiration, glucose is broken down in the mitochondria to release energy, stored as adenosine triphosphate, or ATP. ATP is the energy currency used for most cellular activities. Mitochondria have their own DNA and ribosomes, allowing them to synthesize some of their own proteins. This characteristic supports the endosymbiotic theory. Plastids are unique organelles found only in plant cells. They are as similar to mitochondria as they are double membrane bound and contain their own DNA and ribosomes. Chloroplasts are a type of plastid containing the green pigment chlorophyll, essential for photosynthesis. They are the kitchen of the plant cell. The chloroplast is a double membrane bound organelle. Inside is a semi-fluid substance called the stroma, which contains disc-shaped structures with chlorophyll. Light energy is absorbed here, and sugars are synthesized and stored in the stroma. Mitochondria and plastids, having their own DNA and ribosomes, can make some of their own proteins. These traits resemble certain bacteria, suggesting a shared evolutionary history with these single-celled organisms. Chromoplasts contain yellow, orange, or red pigments, giving flowers and fruits their bright colors. These vibrant colors help attract pollinators and fruit-eating animals for seed dispersal. Leucoplasts lack pigments and are colorless. They are storage organelles, classified by the food material they store, such as starch, oils, or proteins. For instance, leucoplasts in potato and taro store starch. Vacuoles are organelles for storage and support. In mature plant cells, a single large central vacuole is filled with cell sap, storing water, minerals, sugars, and waste. It maintains pressure and keeps the cell firm. In animal cells, vacuoles are sometimes present but are smaller than in plant cells. They provide temporary storage for material.